Hi everyone, my name is Serena,
And my name's Cassandra.
and today we're going to be talking to you about the effects of phosphorus and nitrogen on wetland plant species diversity. So to start, in more general terms, our research and analysis aim to see how nutrients affect species diversity, which in itself is a pretty popular question among ecologists. Most often, this type of research is used to assist with conservation efforts in ecosystems that are threatened, whether that be by human disturbance, environmental tragedy, or the introduction of a new species that negatively impacts the environment, such as an invasive species or the introduction of a disease or parasite. To be able to rebuild these communities, it is imperative to know the living conditions of the species that are present or were once present. In a study by Janssens and colleagues, they investigated the relationship between soil chemical factors and grassland diversity. Within their abstract and introduction, they mentioned that in these threatened communities, most often they have become so rare that the seed bank is insufficient in itself to recreate a plant community. To achieve a degree of restoration, most often sowing of the region is needed in addition to manually recreate soil chemical characteristics to maintain the regrowing community. The chemicals or nutrients that are most important to consider when performing this research are carbon, nitrogen, and phosphorus. However, seeing as the Earth is extremely abundant in terms of carbon resources, it is only important to investigate phosphorus and nitrogen. The specific research question that drove our analysis is what are the effects of varying nutrient concentrations, specifically that of nitrogen and phosphorus, on the diversity of wetland species.
So we thought that our research question would be interesting to study because most of the time when we think about factors that contribute to the growth and biodiversity of plant species, we tend to consider the obvious factors such as basic nutrients and water. However, there is a lot more that actually goes into it, in particular the amount of ion concentrations that are present in these nutrients, which can then determine just how well these plants will grow and essentially if they will survive.
Next, we're going to discuss our method. So the dataset includes 74 wetlands, 37 of which we used to conduct our study. These 37 wetlands, which are considered to be reference wetlands, are considered to be the least disturbed of the complete set as they are free of oil sand tailings and agricultural impacts. These 37 wetlands were divided into 3 regions, of which we examined 2, those being emergent vegetation and wet meadow zones. To provide some context of these regions, emergent vegetation consists of a strip of land preceding a body of water in which plants such as cattails and wild rice grow, either standing above the water or being completely submerged. On the other hand, wet meadows come in many forms, but are often the result of abandoned agricultural fields and previously flooded beaver ponds. This region can be standalone or it may be connected with other wetland complexes, such as marshes. With that in mind, the presence of standing water can occur, however, it is not a requirement of this region. This region has a varied vegetation profile that includes plants such as wildflowers and grasses. Within both regions, sediment chemistry and physical variables were measured, specifically total nitrogen and phosphorus and percent cover of each plant species. To do so accurately, the researchers divided the 2 regions into multiple sites, 79 each, and labeled them accordingly. As for the analytical methods used to analyze this data, we began by plotting the sites of each region against the maximum nutrient concentration, which was provided in the data set. We then calculated Shannon's index, or H prime, which allows for a measure of the diversity of each site. With these diversity values, the site identifiers, and the total nitrogen and phosphorus of both regions, a pivot table was created to condense the material.
Following this, we performed a linear regression for both the emergent vegetation and the wet meadow zones and calculated the relationship between Shannon's diversity and the respective maximum concentrations of phosphorus and nitrogen. The linear regression was done using RStudio and resulted in whether or not the relationships between the 2 variables were significantly increasing or decreasing as a result of the maximum ion concentrations. The R script was able to calculate the estimate value, p-value, and f-statistic. These values were also able to tell us whether or not the variables measured had any significant variations. Next, we plotted Shannon's diversity index against total nitrogen and phosphorus of each site for each zone. In other words, within the emergent vegetation zone, 2 graphs were made, one where Shannon's index is compared with total nitrogen of each site, and the other with the wet meadow zone where Shannon's index and total phosphorus are compared for all sites.
So now we're going to move on to our results. So in this graph, we present the total nitrogen and phosphorus of each site within the emergent vegetation zone. The orange lines represent phosphorus, while the blue lines represent nitrogen. With this, we can see that the limiting nutrient with respect to our two elements of this region is phosphorus, as it is found in the lowest quantity in the majority of our sites. This next graph is the same as the first. However, it presents data for the wet meadow zone. And again, the limiting nutrient of this region with respect to our two elements is phosphorus.
So here we have the linear regression graphs, which we compare the Shannon's diversity of each zone versus the maximum phosphorus concentrations. On the left, we have the emergent vegetation zone, which shows that Shannon's diversity decreased non-significantly with increasing phosphorus. The observed effect size was approximately 0.02, which is relatively small, and it tells us that the variation between our two variables was not very good. On the right, we have the same variables, except we are looking at the wet meadow zone. It shows that Shannon's diversity increased significantly with increasing phosphorus. The observed effect size was calculated to be around 0.05, which again tells us that there is not very much variation between the variables. On this slide, we have the same concept again, but Shannon's diversity is plotted against the maximum nitrogen concentrations. On the left, we have the emergent vegetation zone, which shows that Shannon's diversity decreased non-significantly with increasing nitrogen. The effect size was calculated to be around 0.008, which is very small and tells us that there is little to no variation between the variables. On the right, we have the same variables, except once again, we are looking at the wet meadow zone, and it shows that Shannon's diversity increased significantly with increasing nitrogen. The effect size was calculated to be around 0.04, which is still very small, so there is not that much variation between these variables.
We're now going to move on to the discussion where we compared our results to other research papers. In the Jansen and colleagues study, they found that both nitrogen and phosphorus affected biodiversity. Specifically, they stated that nitrogen is the controlling factor for biodiversity, while phosphorus's effect is indirect due to its control on available nitrogen quantities. In other words, they found that the nutrient most responsible for increasing biodiversity was nitrogen. While in our analysis, we found it to be inconclusive because of all the observed effect sizes, showing very little variation between the Shannon's diversity and the maximum ion concentrations.
In a study conducted in Eurasian wetlands by Venturing Fujita, it was shown that plant species biodiversity may be affected by the nutrient stoichiometry in the environment itself, as opposed to being affected by the individual concentrations of each nutrient. The study suggested that perhaps the plant species are able to adapt to the available environmental nutrients provided. The author hypothesized that the slow-growing plant species reduce their investment in sexual reproduction and seed production because of their shorter flowering periods and longer lifespans. In our results, we found that this could be possibly the case in emergent vegetation plant species. Due to the fact that these species require a high level of water in order to grow, their flowering periods are very short, and thus, because of this, they had less diversity compared to wet meadow plant species, which have a larger range of environmental conditions and can be either in wet or dry environments.
Some improvements for future studies could include a more consistent data set. This particular one technically had 3 environmental zones that could have been compared. However, they utilized a different unit of measure, which could not be converted into percent cover. With that, we could not compare all 3 regions effectively. A second improvement could be to increase the data size, specifically in terms of showing a variety of environments. The researchers narrowed down the data collection to wetlands, so the conclusions drawn could be particular to this environment. We know this is true to an extent, as there have been similar studies with different conclusions, specifically in terms of which nutrient affects diversity. However, there's some studies that do show that phosphorus is a limiting nutrient. Regardless, it would have been interesting to see these comparisons within a single report. Despite these limitations, our study was still worth doing as it showed how diversity is affected by nitrogen and phosphorus in these regions. This information could be applied to other wetlands that are in danger, or it could be applied to the wetlands mentioned in the data set that have been affected by oil tailings and agricultural runoffs, and in efforts to repair them.
Some potential avenues for future research could be investigating the effect of introducing beneficial microbes on a region of plant diversity. We would want to consider questions like, how do they affect nutrient concentrations, and do they affect productivity, and how? Additionally, the study we analyzed could be recreated, but could also look specifically at the significance of micronutrients such as zinc and manganese on plant diversity. Lastly, the effects of introducing leguminous plants into an ecosystem could be studied. So we look at questions like, what would the relationship be between this plant, soil nutrients, and the plant diversity? So that concludes our presentation. We hope you enjoyed it. Thank you.